to be almost nearing the end, but there are a number of excellent talks to come. And before I hand this over to Nella to introduce our speaker for today, I want to remind everybody that next week is the big annual event, the MSc Showcase. And yes, this is what you've all been waiting for. And there's a number of oral presentations from our first-year master's students, and then poster presentations from our second-year master's students. And there are the lucky 13 oral presentations and the lucky 13 poster presentations. And it's a full afternoon. There's an open bar, there's appetizers, and it all starts at 3 o'clock, and it's at the Culinary Arts, the Scratch Cafe. So I expect to see you all there, because it really is a fun event. Now I'm going to pass it over to Nella to introduce our speaker today.
Thank you. Hello, everybody. I am Nella Mora in the Department of Chemistry, and it's my pleasure to introduce the speaker today, Professor Hind Al-Abadleh, a professor in chemistry in the Department of Chemistry and Biochemistry at Wilfrid Laurier University in Waterloo, Ontario. Professor Al-Abadleh is currently on a research sabbatical, and she's at the University of California, Irvine, as the Fulbright Canada Research Chair in Atmospheric Chemistry, Air Quality, and Climate Change. Dr. Al-Abadleh completed her PhD at the University of Ottawa, following Iowa. I had some common aspects to the name. So PhD at the University of Iowa, followed by a postdoctoral fellowship at Northwestern University in Chicago, after which she obtained a position where she has been for the past 13 years? 14. Yeah. Through her research, teaching, and outreach activities, she is passionate about highlighting chemistry roles in explaining environmental problems and transitioning humanity to a more sustainable future. So I hope we'll all enjoy her talk, and the floor is yours.
Thank you. Well, thank you very much, Neela, for the invitation to come and visit Thompson Rivers University, and thank you, Luck, for organizing the logistics of the trip. And I really enjoyed meeting everybody at lunch and the students and learning about all your research. Very exciting. So I'm hoping that I'll give you a taste of the work that I do in my lab at Laurier. My title is very specialized, but I'll be starting with a very general introduction. And then I'm hoping that towards the end, I'll also talk about the atmospheric chemistry work that I do in my lab, which is not highlighted in the title, but we'll get there. Okay. So I wrote a review article when I was a PhD student with my postdoc supervisor that was published in Surface Science Reports. And the focus of that review article was to highlight environmental interfaces in natural and in industrial systems. So here, for example, we're showing a graph that looks at the water-soil interface, groundwater rock interface, and the gas, the atmosphere, and the particles in the atmosphere. So all of these are considered environmental interfaces. And if you look at this table, you will notice that these interfaces are important to study for environmental remediation, for understanding chemistry on particles from an atmospheric chemistry point of view, and also from understanding aqueous geochemistry and the fate of pollutants. And what you want to keep in mind is that the interface is different under these conditions. We were talking about gas-solid, gas-coated solid, or aqueous-solid interfaces. So maybe in your classes, you were exposed to what we call bulk phase chemistry, given that there are 3 phases of matter, the gas, liquid, and solid. But in my research, what I focus on is the chemistry that takes place at the surface, the chemistry that takes place at the gas-solid or the liquid-solid interface, right? I often call that interface region the 4th phase of matter, because to see chemistry at the interface, you need different analytical tools than bulk chemistry tools. And molecules at the interface usually have different properties than they do in the bulk phase. So understanding their behavior becomes really important. So literally, this review article has shaped my academic career because my goal at Laurier was to establish research programs in each of these areas. So in my lab, we have a very active research program in surface geochemistry, where we're interested in looking at the fate of arsenic compounds from their interaction with natural organic matter, phosphorus, and iron as a reactive component in soil. And we've published a number of studies in physical chemistry journals and environmental chemistry journals about how these molecules interact together under environmentally relevant conditions. So I will show you some of that work today. I also have an active research program in atmospheric chemistry that focuses on the chemistry of iron in mineral dust particles, in dust particles that get emitted from anthropogenic activities that include mining and smelting, and how these particles, they interact with a component in the gas phase, in a polluted gas phase, and how atmospheric processing changes the properties of the particles and the gas phase. And most recently, we've received funding from Imperial Oil and INSERC to actually look at environmental remediation of NOx. So this is a new directions of research in my lab that looks at the chemistry of metal organic frameworks. And I don't have data to show yet, but it should come soon. So why do we care about this area of research? So the ultimate goal of working in an area called environmental surface chemistry is to improve the predictive power of models. These models include the climate and cloud chemistry models, earth systems models, pollutant to transport models. So to improve them, we need mechanism-based information that we obtain from field measurements. And in the lab, what we do is that we use field results as the inspiration to design careful experiments so that we understand the trends that people report from field measurements. And most importantly is that theoretical and computational chemistry and theoretical and mathematical modeling contribute to our understanding and interpretation of experimental data through providing atomic and molecular scale information that is hard to access experimentally. So all of these pieces, when you put them together, we can think of the macro scale, going to the micro scale, going to the molecular atomic scale. So I'm hoping to take you on a journey of how the macro scale can motivate research at the micro scale and at the molecular atomic scale. So let's start with the story of arsenic. Arsenic is an element that exists naturally in rocks and minerals and energy deposits around the world. And once it's emitted or liberated from natural activities or from weathering of rocks, it takes on the form arsenate and arsenite. But then field studies have reported the existence of methylated forms of arsenic. Here we're looking at monomethyl arsenic acid. This is dimethyl arsenic acid. And one of the mechanisms that explains methylation of arsenicals is called the Challenger mechanism. And as you can see it here, it involves multi-oxidative oxidation and methylation steps. Arsenic can eventually be lost to the atmosphere through biovolatilization processes that takes place under high concentration of arsenic and under high microbial activities. As for the toxicity of arsenic, you want to keep in mind that the toxicity of arsenic varies with the chemical state and oxidation state. In general, the trivalent forms of arsenic, including the methylated ones, are high toxicity compared to the pentavalent forms of arsenic. Now, current environmental regulations in place impose a maximum contaminant level of arsenic in groundwater and apple juice to 10 ppb, and that's imposed by the USAPA, the World Health Organization, and Health Canada. And in recent years, there have been regulations about the use of arsenicals in poultry feed, so that they were banned in North America and Europe, but they have been still used in countries like India and China. Now, you want to think about the anthropogenic sources of arsenic. So methylated arsenicals and lead arsenate have historically been used as herbicides and pesticides on cotton fields, on golf courses, and in British Columbia, methylated arsenicals have been used as pesticides to combat mountain pine beetles. The disposal of arsenic-containing municipal waste and electronics have also been found to contaminate the groundwater and also to release arsenic into the gas phase above background levels. The industries that emit or liberate arsenic from rocks are mostly mining for gold, copper, lead, and silver, and also the burning of oil and petroleum. So as the demand for energy increases, more arsenic is predicted to be released to the environment from anthropogenic activities. And this is happening at a time when demands for safe drinking water, for safe soil, for agriculture are also increasing. So research into the fate of arsenic compounds is a still active area because of its health and environmental impacts. So if you are in the business of collecting soil samples, what you will see is that soil is chemically heterogeneous and complex. There are mineral components of soil. There is organic matter of soil. Even within the organic matter, you will see different types of classifications. Humus, for example, is classified to have this structure that contain a number of organic functional groups. And these groups are capable of interacting with pollutants and chelating metals. If you look at roots, for example, some roots are capable of releasing low molecular weight organics in order for them to chelate cations that are known to cause toxicity to the plants. So now, if we go now to the micro scale, and if you look at, with a magnifying glass at the mineral-organic interface, you will see that organic molecules interact with mineral surfaces through a number of mechanisms that include ligand exchange, electrostatics, hydrophobic interactions, chelations, cation bridging, and hydrogen bonding. Now, these types of interaction results in changing the properties of the mineral phase: surface charge, surface functional groups, interfacial water structure, and dissolution rates of metal oxides. Where is that useful? Well, all of these properties affect arsenicals retardation in soils. So if you really want to think about what happens when a soil molecule enters, when an arsenic molecule enters the soil or groundwater, you want to think of all these processes. Another way to look at it is that, well, in order to meet environmental regulations, we need to use arsenic removal technologies. In this review paper, the authors have reviewed all the effective and cheap treatment media that remove arsenic from contaminated plumes. And they have found that these media are based on iron, manganese, and on aluminum materials. And they found that the arsenic removal efficiency is dependent on arsenic concentration and speciation, pH, and the presence of other dissolved constituents. Okay? So now, go even deeper. In this simulation, what I'm showing you is how an arsenic molecule in the aqueous phase interacts with a reactive site on a treatment media or on a soil particle. The type of these interactions are driven by thermodynamics, electrostatics, ligand exchange. You want to consider properties like the pKa of the arsenic compound. You want to consider the properties of the surface in terms of its sites. And therefore, you can think of it from a chemistry point of view and simulate important parameters. Now, when we started working in this area, we found books written on the surface chemistry of inorganic forms of arsenic, arsenate and arsenite, using X-ray-based and infrared-based techniques. And the experimental work was coupled with computational chemistry work. And the most important finding from these papers and books was that we still need more data on how methylated forms of arsenic behave in the environment, because the assumption was they behave the same as the inorganic forms of arsenic. So this is the major research question in my lab: how to investigate the effect of organic substitution on arsenate number and type of functional groups on their environmental fate. How do we do that? Well, to answer this question, we need to use techniques that are surface sensitive to probe directly species under environmentally relevant conditions. We want to understand how they bind. What is the structure of these compounds at the surface? How strong they bind. This is the thermodynamics component. And how fast they bind and leave. And this is the kinetics component. We want to also measure directly the heats of adsorption. So this is thermodynamics from experiments. And we want to complement the wet experiments with computational chemistry to aid in the interpretation of the data that we obtain in the lab. So what we did in my lab is actually assemble a team of people, each with different expertise. We have people specializing in using infrared spectroscopy to monitor directly surface species and also to measure heats of adsorption. We have people specializing in density functional theory calculations. We have people specializing in statistical modeling for kinetics. And we have other teams of people specializing in surface complexation modeling to obtain kinetic information. So we studied these types of arsenic compounds, and you can see some have methyl groups, some have aromatic groups. These will have one group, this one will have two. So we are going to focus on arsenate and dimethyl arsenic acid as the two extremes when it comes to their properties and organic substitution. So what we found is that DMA can interact with the reactive components of soil through the formation of what we call outer sphere complexes that are hydrogen bonded, or through monodentate complexes through a single bond to the surface, or through a bidentate complexation that you can see here through the formation of two types of covalent bonds to the surface. Now, the simulations that you see here in this slide are for simulating vibrational modes of bonds in these molecules. So what do we do with that information? Well, we plot calculated frequencies of vibration with experimentally obtained values. And theoretically, you want to get the points to lie on the one-to-one curve. But what you see is that we have good correlation in the low frequency range, but not in the high frequency range. And to explain that, it's just through a scaling factor that we needed to adjust for these values to lie on the one-to-one curve. Why are these important? These are important because these values, these frequency values, when we run the simulation, we can attribute them to different types of surface complexes. We cannot tell that information from the experimental data only. And therefore, once we know that this frequency arises from three types of surface complexes, for example, then the question becomes, if we have simultaneous formation of more than one type of surface complex, and therefore the question is, but relatively speaking, do we have more of this versus that? How can we gauge the abundance of one surface complex versus the other? Now, for arsenate that has no organic functional groups, when we did the same studies, this is the experimental data we obtained, and this is the theoretical data we have calculated for neutral surface complexes versus positively charged surface complexes versus negatively charged surface complexes. And when we compare the frequency values with the ones that we obtained experimentally, what we concluded is that as a function of pH, we have the negatively charged complex dominant under basic conditions, and the neutral complex dominated under neutral, relatively speaking, conditions, and the positively charged is dominant under acidic conditions. Okay? So now, good, but now the question is, if we have multiple types of surface complexes, are they all bind with the same strength? And this is where we get to calculate Gibbs free energy and enthalpy, and I'm sure most of you have taken that in your general chemistry classes. You know, the Gibbs energy products minus reactants. So this is the reaction that we're concerned with, a reactant in the aqueous phase interacting with the surface, and the formation of a cluster that releases water in the ligand exchange process. So this is the data that we have found. On the y-axis here, I'm showing the difference in Gibbs energy which tells us the favorability of a chemical reaction, or the spontaneity of a chemical reaction, relative to the outer sphere. And what you see is that the formation of a monodentate versus a bidentate is favorable, but then the formation of this complex has a very high energy barrier. So the conclusion that experimentally we expect to see these types of surface complexes, because these will require lower energy barrier to form than the formation of the bidentate complex. Okay? Now the case for arsenate is different. Arsenate it is thermodynamically favorable to form monodentate and bidentate complexes. But then the transition from monodentate to bidentate has a lower activation barrier, and it's a complex barrier because of the hydrogen bond breaking and formation. So we concluded that it is this bidentate complex, the most thermodynamically stable complex that forms, which has been observed experimentally using infrared and X-ray based techniques. So what's the conclusion so far is that computational chemistry and comparisons with spectra of species in the bulk are very useful in structural determination and also predicting thermodynamic favorability of surface complex formation. Now, as the mathematical modeling, and I know I've heard that some people are involved in modeling studies, we actually do mathematical modeling on the systems that I've shown you as well. But then when you deal with models, it's a tricky business, because some models are empirical. They're only valid when you have defined conditions, and if you want to predict, then usually you have to redo the experiment again. But there are other types of models that incorporate information about your system, such as surface species, chemical reactions, mass and charge balances, and surface activity coefficients, and the consideration of charge on both the reactant and the surface. When these models incorporate these variables, then these models become able to predict future behavior of a chemical reaction without you redoing the experiment. Okay? So these are the types of models you really want to work with. So the model that we wanted to work with is called the triple layer surface complexation model. This model divides the surface or the interface into three regions: the region closest to the surface versus the one that is right above it versus the diffuse layer close to the bulk. And here we have inner sphere versus outer sphere complexes. And as we've learned from general chemistry, the equilibrium constant for a reaction at the surface will be related to the surface potential, and a surface potential will arise from the fact that we have charged species at the in-surface and also in the aqueous phase. So for the reaction of DMA with iron oxides, these two reactions were the reactions that we needed to have equilibrium constants for thermodynamic information. And the model results as a function of concentration and as a function of pH that you see here in red give us a very nice fit to the experimental data, using two equations that suggest the formation of these two complexes. And these two complexes were initially obtained from spectroscopic data and computational chemistry data. So the story from thermodynamics is that DMA forms these types of surface complexes with these thermodynamic parameters, versus arsenate forms this complex with this thermodynamic constant. See, you can see that arsenate with this very high value interacts stronger with iron oxides than DMA, which interacts weaker. Now that has implications, as I'm going to show you later. Now, of course, I have shown you result up to now on an idealized surface. In reality, most metal oxide surfaces have organic molecules on them. So we decided to work with these simple organic molecules to model natural organic matter. And what we found is that these organic compounds interact with iron oxides in this fashion. And the conclusion was that surface oxalate and citrate flip the charge of a hematite surface from positive to negative because of their carboxylate groups. and that oxalate and pyrocatechol are the two extremes in modifying surface charge and the type of surface complex. Now, what does that do? Well, if you have a chemical reaction, when you have citrate, oxalate, and pyrocatechol on the surface, and you have these species in the aqueous phase, look what will happen when you look at the desorption. Desorption is the process by which these organic molecules leave the hematite surface. Like they basically, you're competing for the same site, so something has to leave. So in this graph, we're showing the speed by which these chemical species in the aqueous phase remove organics from a hematite surface. So what is the conclusion? Look here, is that the rate by which arsenate removes citrate is almost 3 times faster than chloride. In the case of pyrocatechol, also arsenate removes pyrocatechol 3 times faster than chloride. And in the case of oxalate, arsenate removes oxalate 5 times faster than DNA. So what does that mean? It means that if you are in an environment where you have high concentration of organics and you have arsenate around, arsenate will be able to displace these organics from the mineral surface and instead interact directly with that mineral surface. So that has consequences on the retention of organic matter on minerals and soils. So, so what are the implications and the summary so far? Increasing organic substitution in arsenate increases the number of weakly bonded complexes, and that has consequences on their mobility and bioavailability. Iron oxide materials can remove organic forms of arsenic in the absence of competing species, and because of their high affinity to arsenic compounds, iron oxide nanomaterials can act as vehicles, right, for transporting pollutants. When you have organics at the interface, low molecular weight organics can enhance or inhibit arsenicals binding, and that the reaction of organic matter with hematite largely depends on the type and the concentration of the desorbing species. This is basically what I was telling you about. And mechanistic understanding can lead to more accurate modeling and guidance to designing remediation technologies. Because if you have a water sample that has different forms of arsenic in it, and you are going to clean that water, but then each of these arsenic species interacts differently with your removal material, then you want to know, if you pass that polluted water through that material, are you going to have 100% removal efficiency? Maybe you will for some form of arsenic, but not for the other form of arsenic. That's the bottom line, is that when pollutants interact differently with materials, then you have to be aware of their removal efficiency. And what I've showed you is just data that supports that, right? And that's in an ideal system. If your system has organics in it, then you will have competition. Which of these species will win the competition, right? So these are the types of questions we ask. So I finished the geochemistry component of my talk, and I want to switch gears and talk about atmospheric chemistry research now. But before I do that, maybe let's have a discussion or questions from the floor about the material that I presented so far. Okay, so the question is, what is the most stable form of arsenic, and what is the one species that is available for plant uptake? The answer is, you really want to analyze the microbial activity in that environment. If you have high microbial activity, then you would expect to see methylated forms of arsenic alongside the inorganic forms of arsenic, as well as you want to analyze the oxidative properties of your soil and redox properties of your soil and the pH of the soil, because species of, as you have seen, these arsenic species are acidic, so are acids. So their protonation state gets affected. The oxidative potential of the soil will affect the oxidation state of the arsenic species. In general, the deeper you go in soil, the more reductive the environment becomes, And you could see more of the oxidation state of 3 than 5. The higher you up in the soil layers, the higher the higher concentration of the oxidation, the higher oxidation state, which is oxidation state of 5, is what you see. It is less toxic. It binds stronger to iron oxides. The reduced form of arsenic compounds have no affinity to iron oxides. That's why they're more mobile. So if you look at, so if you really want to, then as for plants, the question about plants, well, it turns out if plants can have iron centers in their root structure, this is what will have the highest affinity to the oxidation state of 5 of arsenic compounds. Yeah. So you want to analyze the root structure from the point of view of the metals. What are the metals in the root structure that could take up and bind arsenic compounds strongly? And in addition to the diffusion properties of those roots. Yeah. Yeah. Yes.
You showed some data with the change in the present.
Yes.
Are you able to calculate the change in entropy and the change in enthalpy?
Yes, we did that in the paper we published. And I don't have that data here, but we found a negative change in enthalpy, so the reactions were exothermic, and the entropy change was positive because of the change in the water structure near the surface. So the absorption of arsenic compounds and the displacement of water, it creates a more disordered environment than the initial environment. Yep. Okay? Yes?
Is the arsenic present in oil sands a different form than afterwards when it's processed in industry? Is that a different form?
This is a very good question. The question is the arsenic in the oil sands is in a different form than it is after processing? Yes, absolutely. Because in the oil sands, you have a high concentration of the organics. You will see, and the environment in which the oil sands have developed have allowed for reactions that will form aromatic forms of arsenic, for example, or methylated forms of arsenic. And then the processing part, with increasing heat, could break down the organics, and then you end up forming the inorganic forms of arsenic, by all means. So that's why speciation information is important when you analyze oil samples, oil sand samples, versus before and after processing, to see what does that process have done to the actual species. But yes. Okay, other questions? Okay, let's move on. Okay, let's move on. Okay, now, so as I told you, we also have a productive research program in atmospheric chemistry, and the main motivation for us to work in this area is by the data that is published by the IPCC report. And this is a picture that has been taken from their latest assessment report. And it basically shows the contribution of greenhouse gases to radiative forcing. So radiative forcing is basically this net energy parameter that helps us understand cooling versus heating of the atmosphere. So positive radiative forcing means warming of the atmosphere. Negative radiating forcing means cooling of the atmosphere. And all of these values are relative to pre-industrial values. Okay? So what this figure shows is that we pretty much understand how greenhouse gases operate, right? The long-lived, well-mixed greenhouse gases versus the short-lived greenhouse gases. And because we understand that very well from a scientific point of view, that's why the error bars are relatively small, okay? Because the size of the error bar determines the current level of scientific understanding. Now, if we look at other components in the atmosphere that contribute to climate change, those are aerosols and their precursors. And then look at the relative size of these error bars. Yeah? So the size of the error bars is higher for the aerosols. They could have a cooling effect. Some will have a warming effect. And if you compare these anthropogenic contributors to climate change with natural forces, you will see that the natural contributions are well understood, but are actually smaller than the anthropogenic sources from particles and gases. And then the bottom part of this figure compares the fact that over time we have seen warming happening as a result of human activities. But then our job now as scientists in this field is to actually make sure that we reduce the uncertainty in this component of the atmosphere that impacts climate change. So let's focus on aerosols and their effect on the climate. Aerosols affect the climate directly through absorption and scattering of solar radiation, and indirectly. So indirect effects of aerosols include health effects because of inhalation problems, visibility reduction as a result of light scattering. Aerosols also provide surfaces in which chemistry that can take place, right? These little particles in the atmosphere, these are little reactors, chemical reactors. They don't stay the same, and they transform, they get to transform, and they transform the atmosphere around them, okay? And then, as a result, aerosol-cloud interactions, when it comes to formation and lifetime, also get affected. And when you affect the formation and lifetime of clouds, what gets affected? Precipitation rates. That's why one of the predictions of the impact of climate change is that precipitation will increase in some areas, and droughts will happen in other areas because the cloud structure will be different because of the amount of particles and the chemistry of these particles. Okay? So if you look at the life cycle of aerosols in the atmosphere, this figure shows emissions of primary particles from arid regions, forests, urban centers, and coastal regions with their aging, which means chemical transformation, and also the nucleation and formation of new particles in the atmosphere. We talked about how aerosols affect the climate, and eventually, depending on their size, some of these particles get deposited on lakes and trees and land. But then all of these processes that you see in red require molecular level understanding in order for us to improve the predictive power of models used by regulators. Okay? And this is where research comes in. My group is interested in the metallic component of primary particles. There are field campaigns that collect particles from these emission sources, and they have reported the presence of transition metals such as iron in these particles. So if we look again at the microscale, molecular level scale, this is a schematic of a diagram that looks at the chemistry of hematite or iron species in general that could take place in a cloud droplet. Yeah? So we know that, for example, iron species can oxidize sulfate, bisulfite to sulfate. We know that hydroxyl radicals can oxidize water-soluble organic compounds. We also know that there are some ligands like sulfate and carboxylates that can bind and complex to iron, and then the photolysis of these complexes can create reactive species that can carry on versus reactions. In fact, this chemistry that you see here is the chemistry that helped us explain acid rain in some of the regions. And another important factor is the fact if you emit solid iron oxide, with time and with chemical reactions, you can liberate that iron into the aqueous phase. What else field campaigns measure? Well, field campaigns measure the pH, or the acidity of these aerosols. So here we're showing some of the chemical components that actually affect the acidity of the aerosols. And the pH scale that you see here was calculated based on a thermodynamic model that incorporates the type and the concentration of these chemical species. And these are the field campaigns that have been conducted in China and in North America. And why are these graphs useful? Because they can help us design experiments in the lab with model compounds that have iron in them. So in my lab, we work in this pH range between 3 and 5, which basically overlaps with some of the acidity that have been reported for some of the field campaigns. So now this is a schematic that I want you to examine. What we know is that the chemistry that takes place in a droplet of water versus that takes place in a tiny particle of water is going to be different, because it's a function of the aerosol liquid water content, it's a function of temperature, it's a function of pH. And all of these factors come into play when we are trying to understand the chemistry of these systems. So in my lab, we use iron materials that provide insoluble source of iron or soluble source of iron. And we use these organic compounds that have been identified in real aerosols from field campaigns. Guaiacol, pyrocatechol, or dicarboxylic acids that you can see here as well. Okay. So let me show you a couple cases of the chemistry of iron. This is a reaction between catechol and iron chloride. Initially, you see a transparent solution, but once you mix these solutions together, you end up forming a greenish solution, and then that develops into a black solution. And the UV-vis spectra of this solution shows very distinct peaks in the UV-vis spectra at 715 and 390. And then you also see that we have particles that take place in filters. Now, these particles are similar in properties to soot particles. You know, soot that you obtain from burning fossil fuels, from biomass burning. They're tiny agglomerates of black carbon. These particles that we formed without fire, I call them fireless soot, fireless soot. They're black. This means that they can absorb radiation. If they can absorb radiation, then they can carry on photochemistry. So a whole window of chemical reactivity opens up from particles that form without igniting any fire. So that was a very interesting finding, and we wanted to confirm that if we use an insoluble form of iron, such as oxide, and we try to simulate what will happen to this if it got emitted into the atmosphere and reacted with acidic gases. So how did we do that? We reacted hematite in an acid for 10 days. We filtered the unreacted hematite, and then we adjusted the pH. We reacted that with catechol, and we had the particles form right away. And also we did the same thing with the guaiacol, and we ended up forming brown particles. So this type of simulation was important because the hematite is a very reactive component of mineral dust particles and of iron dust particles that get emitted from anthropogenic sources. So the fact that if hematite reacts with acids, it can carry on chemistry like that was important to report and to talk about. Okay. In other studies, we changed the organic compound. This is a fumarate of transparent solution, but when you mix it with iron, you end up with an orange-like solution, and when you filter, you get particles that contain about 27% iron. Now, the UV-vis spectra of the solution before and after filtration showed that these shifts in the UV-vis spectra are due to the particles. And this is important, again, because they can influence photochemistry in the atmosphere. Why is this work important? Well, because there is something called secondary production of particles that can absorb light. And if these particles can have a property similar to black carbon or the particles that are obtained from biomass burning aerosols, then this is a pathway that is catalyzed by metals that we should really incorporate into models, because this type of pathway is not there in the models yet. Another implication is on the chemical reactivity of multi-component atmospheric particles. If you have these particles that have iron in them, they have organics, that the acidity of these particles are different, then you can expect a suite of chemical reactions to take place. And that can change the properties of the particles. And all of these processes need to be incorporated into cloud chemistry models for better simulating the transformation of aerosols in the atmosphere. So basically, to sum up what I have told you, this is the lifetime or the life cycle of an iron particle. Once it gets emitted from primary sources, it can undergo all these processes. But the data that I have shown you tells us that we can actually form particles. We emit particles, we react them with these compounds in the gas phase, and all of this is happening, could happen in the dark or in the light. But what I've showed you is that under dark conditions, you can form particles from reactions, and that these particles, we still don't understand how they actually react with pollutants in the gas phase and how they contribute to cloud formation and lifetime and chemistry. Another thing is that we don't understand how the photochemistry of these particles is, right? Because they've seen these particles are colored, and therefore they can absorb solar radiation. So all of this work is currently being investigated in my lab as we speak. And of course, you know, I've been blessed by a number of hardworking students who helped me run the work. So I always say that none of the work I showed you is mine, it's my students' work. So I'm grateful for their handwork in the lab and their interest in the fields of geochemistry and atmospheric chemistry, and also my collaborators who have contributed to help with their technical expertise in instrumentation and mechanistic information as well. And also, thank you for listening to my talk, and I welcome your questions.
Just wondering if the size of these nanoparticles, does that affect how they react and affect properties, obviously?
That's right. So I have backup slides on that. Let me show you. We did dynamic light scattering measurements. Okay, where are you? Size, particle size. Where are you? Is it taken away? Okay, here. Okay. Not this. Yeah, I'm giving this talk tomorrow at UBC. So, but it will focus mostly on the atmospheric work. Let's see, it should be here somewhere. So the dynamic light scattering show that they are in the nanometer size. Here we are, okay. Here. Okay, so this is the polycatechol, polyglycol data. Initially, the nucleation happens below a micron, and then they grow to a micron size around about 40 minutes. And then the growth rate is different for these chemicals. As for the other ones, the organometallic polymers, so the organometallic polymers, so this is iron polyfumarate, for example, and this is in nanometers the scale. So you can see that the initial nucleation is in the 100 nanometer size range. And pH and the ratio to the iron actually affects the particle size with time. But they quickly grow to micron size. They start in the nanometer and they grow. Yeah. Any other questions? Yes.
Let me go back to the first part of your talk. When you're able to calculate the thermodynamic parameters,
Yes.
are you able to use that to determine the type of manifestation?
Yes. So that's, yes, here. Okay. Where are we? Here. So, here. Yes. So basically, we constructed this plot after calculating the Gibbs free energy for the formation of outer sphere, monodentate, and bidentate complexes. And we calculated the Gibbs free energy for these clusters with different charge, like a neutral cluster versus a positive cluster versus a negative cluster. And in all cases, Gibbs free energy was negative. So these reactions were spontaneous. But what we were hoping to show in this graph by taking the difference in Gibbs energy relative to the outer sphere is that the monodentate and the bidentate are more thermodynamically favorable than the outer sphere. But what do we observe experimentally? The data that we have showed that we actually do not have the bidentate experimentally. What we have is outer sphere and monodentate. So that motivated the work to look at the energy barrier of transition between the monodentate and the bidentate to help to explain the experimental data. And it turns out that we have a very large energy barrier to go from the monodentate to the bidentate, but we don't have that high barrier in going from outer sphere to monodentate. So we concluded that it is these two that we observe experimentally. And we actually leaning towards the abundance of outer sphere more than the monodentate. But we have evidence that we have some inner sphere complexation in addition to the outer sphere. Yes. Okay.
Thank you once again, Hind,
My
for coming
pleasure.
and sharing all of your work with us.
My pleasure. Thank you.
Thank you.
Thank you.